Hi, I'm Judy Cole, the Executive Vice President and CEO of the MIT Alumni Association, and I'm delighted to welcome you to this web production of the MIT Alumni Association.
Welcome to the MIT Faculty Forum Alumni Edition. I am Ann Gibbons. I'm a writer for Science Magazine, and I was a Knight Science Journalism Fellow. This is an interactive forum, so we encourage your questions. Use the Google form below the live video feed to ask a question, or you can tweet one to MIT Better World. We'll get to as many questions as we can. So this month's topic is CRISPR, the CRISPR revolution, or advances in gene editing. So biologists continue to hone their tools for deleting, replacing, or otherwise editing DNA. And they're using a strategy called CRISPR that has become one of the most popular ways to do genomic engineering. By using a molecular method that bacteria usually use to fight off viruses, by snipping some of their DNA, researchers have been able to adapt this so-called CRISPR system to edit genes in many species, ranging from yeast and bacteria to fruit flies, and even, in 2 cases, human embryos. The unprecedented control over genes has allowed many new types of experiments, but also obviously raised questions about what CRISPR can enable and the ethical issues that come with that. At least one group has already used CRISPR on human embryos, sparking calls for a moratorium on similar work and an international summit at the end of last year to discuss the science and ethics of human gene editing. Meanwhile, CRISPR is making it much easier to generate genetically modified animals and plants, creating new regulatory issues that scientists, agencies, politicians, and ultimately society must address. So last night, just for the fun of it, I Googled CRISPR, and here's what I read. These are headlines just posted in the last 24 hours. One was, first human test of CRISPR proposed. That was in the MIT Tech Review. The second was gene editing. CRISPR blocks cancer growth. That was in Nature. Another was, virus hacks host genome, steals CRISPR to protect itself. So today our alumni panel will help us separate myth from reality, share their current research with us, and perhaps untangle some of the hysteria around the latest headlines. Panelists, let's have you introduce yourselves and give us an overview of your current research on the state of gene editing in 2016. And we're going to start with Melissa Harrison, who will also give us a brief definition of CRISPR. So let's start with Melissa Harrison introducing herself, and then we will go to our other 2 panelists, who are James Dahlman and Keith Tayo.
Hi, everyone. So I'm in the Department of Biomolecular Chemistry at the University of Wisconsin, and I'll just give some brief visuals to go along with my introduction. So essentially what my lab is interested in is how you can take 2 specialized cell types, in this case a sperm and an egg, and unite them to create a new organism. And ultimately what happens when these 2 cell types come together is they make a single cell, and this single cell now has to divide and differentiate into all of the cells of an adult organism. And so the DNA in this single cell and in all the cells of the adult are the same, so we're really interested in how is this DNA, this genome, interpreted to drive development. And because there are so many shared properties amongst all organisms, we do our studies in the fruit fly Drosophila melanogaster. And CRISPR-mediated genome editing has been incredibly influential for us to allow us to introduce specific mutations into the Drosophila backbone. So high-throughput sequencing has identified numerous DNA sequences that are associated with diseases such as cancer, but it's really essential to determine whether those changes in the DNA are actually causing the disease. And so what CRISPR has allowed people to do is to make those exact mutations that are associated with disease in any organism that's a good model, and to test what the effects are of that mutation. So CRISPR has become really facile and quite easy, and so these are a number of organisms that people have already modified using the CRISPR-Cas9 system. So that's, for me, why it's such a useful system, but just briefly, what is so novel and how does it work? So it's important to keep in mind that what really the CRISPR system is doing is creating a double-strand break in the DNA. And that's really all that it's doing. It's the cell's job to repair that DNA and the researcher's job to control how that DNA is being repaired. So there were tail nucleases and zinc finger nucleases that could cut double-stranded DNA and make this break. But what was revolutionary about the CRISPR system was how easy it was to generate these breaks in a much more targeted and specific manner. So where the researcher steps in is both the introduction of the programming of this Cas9 protein, but also researcher-directed strategies to influence the cellular repair machinery. So briefly, what are the basic components of CRISPR-mediated gene editing? One is the Cas9 protein. This is the nuclease that cuts the DNA, and the other is a small guide RNA that easily programs this protein where to go in the genome and how to cut it. And then, if wanted, the researcher can provide a template for repair. And so a real brief example from our lab is that we wanted to look at where a protein is in the cell. And so we can add a fluorescent tag to that protein by changing the DNA. So here's say the original gene. What did we do? We programmed that Cas9 to make a double-stranded break, and we gave it a repair template that had that fluorescent tag. Then we could screen and identify flies that had this modified genome, and we could find that we could now visualize this protein in the nucleus of a developing organism and see where it went. And so I just want to be very clear that this was all part of an ongoing collaboration with two other members of the UW-Madison community, Jill Wildarner in chemistry and Cato Chiner-Giles in genetics. And we have a website for anyone who's interested. And then also this is my website if people are more interested in my work. So, so with that, I need to get out of screen sharing.
So let's go to James. Could you introduce yourself and tell us how you're using CRISPR?
Sure. So, hi everybody. My name is James Dahlman. I'm a recently appointed assistant professor in the Department of Biomedical Engineering at Georgia Tech. And we use CRISPR a few different ways. My background is actually in drug delivery and nanoparticle design. So we do a lot of targeted drug delivery of CRISPR therapeutics. But I also have some background in actually engineering and designing the CRISPR system itself. And I did this as a postdoctoral fellow while I was at the Broad Institute studying with Feng Zhang. And there we learned a pretty important concept, which is, well, I guess we're not the ones that learned it, but it's an important concept to point out. It's this idea that the CRISPR-Cas9 system, as Melissa said so well, is a really effective and useful system. And one point I'd really like to highlight is that when you think about the system from an engineering perspective, it's a really nice system to work with because you can engineer both the protein itself, so you can actually engineer that Cas9 protein to do other stuff that you want to do. You can have it turn on a gene instead of turning off a gene. You can make it cut, one strand of DNA but not the other. You can make it cut no strands of DNA. So you can actually engineer the Cas9 protein to do other things. But that's not all, actually. A second really big concept, and one of the reasons why this technology has been so useful, is we can also engineer the guide RNA itself. And so to give you a brief example of this, we were able to publish a paper last year where we showed that by changing the structure and the size of the guide RNA, we could actually have it target the correct genomic DNA, but not cut the DNA, and instead actually turn on gene expression with a Cas9 protein that normally deletes the gene. And so if you think about this, what we're able to do is use this special designed RNA to upregulate gene A in a cell, and then put in a regular guide RNA to cut gene B in a cell. And so we call it orthogonal gene editing, which colloquially can sort of be viewed as this. So in the same cell, we can turn off gene A and turn on gene B at the same time. And this is done by basically engineering the RNA. So we do, like I said, we'll talk about this a little bit later, but I'm interested in the sort of therapeutics and translation of different genetic therapies, including CRISPR. But the real take-home point is, if you guys are engineers listening, this is a really engineerable system, and there are many, many examples of engineering both the protein as well as the guide RNA to do different things.
That's great. Let's go to Keith. Could you please introduce yourself and tell us how you're interested in or using CRISPR?
Sure. My name's Keith Tayo. I'm an assistant professor at Northwestern University. I'm in the Department of Chemical and Biological Engineering. Let's see, I have some slides I'd love to share if I can remember the process here. Let's see. Okay, you guys can see that?
Yes.
So I'm interested in engineering microbes, so things like E. coli and things like yeast that really take advantage of many of the things that biology does really, really well, and trying to convert those into something that's generally useful for society. And so those two things, as a chemical engineer, I'm really interested in is catalysis, and as you think broadly, you know, the exquisite chemistry that happens inside a cell, where you take something like a sugar and you make these extremely complex polymers and other types of molecular structures is really amazing, and so harnessing that is a big interest of mine. And the second thing that nature does very well is sensing, and so whether that's running from something or running towards something, we've all evolved very sophisticated ways of detecting our environment. So that's something else that I'm interested in, co-opting some of these natural processes to do something useful for society. So I look at that in a couple different ways. So as a chemical engineer, we'd like to be able to synthesize chemicals that we couldn't make otherwise. So some of these are drug compounds that are inefficient to be synthesized by traditional organic synthesis routes, biofuels, other high-value chemicals that require a lot of land if we were to grow crops that make that chemical, but we could make it very, very efficiently in a bioreactor. So example there would be like essential oils, or it takes a ton of plant to make a small amount of essential oil. On the other side of things, we think about yeast sensors, and this is really, I think my next, we'll skip to that slide, is how do we take yeast and make it into something that might be useful as a healthcare diagnostic. And so yeast are very good at sensing their environment, because you can make them dry active, then they're cheap and transportable. So in developing countries, this could be a very valuable way to diagnose diseases in places where there may not be reliable electricity or the type of hospital we enjoy in the United States. And so at the end of the day, you could imagine something that looks like a pregnancy test, but where that strip is now a yeast cell, and it makes things interesting. And then finally, this is just emphasizing that idea of novel reactions that we can carry out. So paraxenomide in the top half of this is a tuberculosis drug that's currently made by chemical synthesis, and what I'm showing here is a reaction diagram where we use enzymes in non-traditional ways to get to those products. And so where CRISPR has really enabled what I do is based on this design, build, test, learn cycle, which is what we follow in synthetic biology. And so if you think of design and learn being the sort of computational aspects of understanding complex systems, the build is often are trying to put together genes from different organisms, modified genes in that yeast or E. coli cell that we're trying to work in. And I have there in 2010, that was when I was doing my postdoc, but even you know, a year and a half ago in my lab, you know, depending on the type of modification we'd want to make, that could be up to 1 per month is sort of the slow rate that would happen. And basically now 4 per week is very, very doable, and if we get more creative, we can actually be even faster than that. But as you can imagine, what that allows us to do is learn much faster, because this what used to be rate-limiting step in our ability to understand these systems, now because CRISPR is so simple and facile to use, that build step has extremely compressed in terms of time, and so we can now go through this cycle really at like a once per week type of rate.
Very interesting. Okay, so I'm going to start taking questions. Can you guys hear me? And I'm going to start with James, but first I want him to explain one thing. When you were talking about working with gene regulation and engineering these Cas9 proteins, what organisms are you working in? What's your model organism? And then after you answer that, I want you to take Adlai's question from Cambridge. And here's his question. Have you had success with delivering gene editing tools, CRISPR and some others, beyond the blood-brain barrier for targeting certain neuron types or aberrant genes?
Great. So that's number one. That's a great question, and I'll get to that in a second, as suggested. But so to address the first question, which again is which model organisms are sort of commonly used, this answer is probably variable from lab to lab. But I know that in many laboratories that I'm familiar with, we perform a lot of editing experiments in human cells, so we don't really have an organism, it's just a cell at that point. But you can also perform editing in mice. I've heard rumors of people doing it in larger animals as well, non-human larger animals. And actually, as far as I remember reading something at some point a few months ago saying that basically every model organism that this has been tried in so far, it's been successful or something like that. I don't know if that's 100% of the case or not, but I've heard, of work going on in C. elegans, certainly in bacteria, in mice, in rats, and as we heard just a few minutes ago, also in flies as well. So we can use a lot of different model organisms, and one of the, I'll just say something quickly related to that. One of the things that's kind of interesting and perhaps unexpected is keep in mind that this system, this CRISPR system, is a system that we've basically hijacked from bacteria. So this system wasn't really designed, if you think about it, to work in mice. But the system is so robust, even though it was evolved in bacteria, that we can basically take it out of bacteria and try it in all these different organisms. And so it's a really powerful system, and that's not a given. I just want to make that point. Now, moving on to the second question related to the delivery specifically across the blood-brain barrier, as far as I'm aware, that has not been done systemically. But we also have to keep in mind that with the brain, there are other ways to access the brain. So when we think about delivering a drug to the brain, there are a few different avenues where you can inject the drug. So as an example, you can take a pill and try to get something into the brain that way. You can inject it basically through the equivalent of an IV bag and goes straight into the bloodstream. But with the brain, you can also try to inject it directly into the brain by performing a little microsurgery. And that has been done, actually. People have done that little microsurgery directly in the brain. But if you're asking about sort of what we call systemic delivery, which is drug delivery, again, where you're trying to basically inject into a vein and get it to go to the brain or the heart or the lung or really anywhere you want to target it after injecting into the brain. As far as I know, that delivery has still been extremely challenging. And we can go, if people are interested, we can talk a little bit later about why that's the case. But the short version is that delivering CRISPR-Cas9 therapeutically has been a huge challenge and will probably remain a huge challenge, and therefore a big focus of research for several years. So to repeat back and to state the big bullet points, we use it in a lot of different model organisms, number one. Number two, when we think about the brain, people have performed gene editing by spraying stuff locally into the brain after performing a little surgery, but people have not successfully delivered CRISPR-Cas9 systemically by injecting it into a vein or putting it into a pill. And actually, people haven't done that for other organs as well. It's been very hard to get systemic drug delivery for other organs.
Can you say, James, why it has been so hard to get systemic CRISPR-Cas9 drugs? What is going on there? Is it a regulatory issue? And technically, how is it hard? How close are you to developing, how close are people to developing any kinds of drugs or therapeutics?
Yeah, that's a great question. And the reason why it's been difficult is because the system is a complicated system from a drug delivery standpoint. So I will give you an example. When you want to deliver a regular drug, let's say you want to deliver cisplatin, which is a chemotherapeutic, or you want to deliver what's called an siRNA, which is a very small naturally occurring RNA. That drug delivery is already hard enough. So if you want to deliver the chemotherapeutic, you have to inject the drug. It has to avoid getting cleared out by the kidney, by the liver, by the immune system. It has to get to the right tissue. It has to get into the right cell. It has to get into the endosome of a cell, out into the cytoplasm of a cell. All of these steps, even with a very simple drug like a chemotherapeutic, which is just one simple molecule, is really hard. Now, you look at CRISPR-Cas9. You have all those hurdles still. But in addition to those hurdles, you also have the fact that the system requires both a protein and an RNA. The protein and the RNA have to come together, and then the protein and RNA has to get into the nucleus for it to work. And so those additional steps make it really, really inefficient. So it's a complicated system biologically, and because drug delivery is already hard with simple molecules, drug delivery gets really, really hard when it's a complex molecule like CRISPR-Cas9.
We have a question that comes from Mount Holyoke, and that is a question from Al in Mount Holyoke. How much of your work is coding, and I imagine that means modeling, versus how much of your work is in the lab in Petri dishes versus wet lab kind of work? So Keith, maybe you can take that question.
I'd be happy to. As I was alluding to in that circular diagram, sort of the left half of that was basically coding, computational analysis, and in the past, the amount of data we could generate was relatively small, so the amount of analysis we could do was accordingly small. What CRISPR has really done is by greatly increasing the number of hypotheses that we can test, allows us to generate much, much more data, and so increasingly now computation is a very large part of what we do. 40% of my lab, so 4 of 10 graduate students are 100% computational, and are really just trying to take the really amazing data that we're getting now and trying to answer larger, more systemic questions that would just have been impossible a short time ago.
So I have my own question for Melissa. When you talk about using CRISPR-Cas9 to study development, what is most exciting for you right now in using this system to study development, either in your lab or other labs? What do you see as the potential there, and what is underway that is particularly promising or exciting for you?
The possibilities are endless, and I can't limit it to one. So one of the things that's really interesting to us is that sort of what I alluded to is that there are these mutations which are associated with disease. So cancer genomics has really made a huge impact in sort of sequencing large numbers of cancers, finding mutations that are associated with them, but really understanding what those mutations do and whether or not they're causative for any certain disease. And so now what we can do is actually make that exact mutation in an organism. And for most organisms, homologous recombination hadn't been facile enough. Even in Drosophila, it was incredibly hard. And so the fact that we can now go through and within a matter of a month make a specific disease-causing mutation in a fly and use that as a model, or in other organisms, mice, axolotl, swine, whatever. So that's one really exciting thing. And then the other is the fact that we can now label proteins in vivo in their endogenous setting and study what they're doing. So with the fluorescently tagged version, we're actually watching it over cell divisions and seeing where that protein goes and seeing where it moves over development and what cells it's being expressed in. So really, for us, it's an amazingly powerful tool to probe at the genome level what is going on. So.
And what are you seeing as far as that protein over time? What is particularly interesting about the system you're looking at?
So this protein actually reprograms the embryonic genome to totipotency in Drosophila, and we think similar things are happening in mammalian cells. And so what we can see with this this protein is that it basically behaves in ways that we might or might not expect and predict based on its dynamics.
So we have another question here. This is for James now. Jose in San Francisco wants to know, where do you see these kinds of therapeutics 10 years from now? Over-the-counter deliverables? Can you answer that, James?
Sure. So, Jose, thank you very much for the question. It's another great question. So I'll answer the second part of your question first directly. I have to be honest, I don't think it's likely that in 10 years we'll have over-the-counter versions of these things, simply because the implications of these gene editing therapeutics are really, they're serious implications, and when you're going in and making, permanent changes to DNA, that's a pretty serious thing. And so the development of therapeutics is going to be a little bit more specialized, I think. And I can give you a few examples of a few therapeutics that may be reasonable within, let's say, a decade. And this is, please don't ever quote me on this, because developing a drug takes a really long time. So actually, instead of saying 10 years, what I'll say is I'll describe some therapeutics that are likely to perhaps come out first, instead of putting a timeline on it. One of those therapeutics might be something related to the eye. So as we talked about a minute ago, delivery is a really big problem. And so if you think about the eye, you can literally take a drug, put it into a needle, and inject it directly in your eye. Many drugs are actually administered that way now. And so there are going to be CRISPR-based therapeutics that people will try to advance, where you try to go in and edit or delete or manipulate genes that cause, for instance, like a progressive blindness. That's a genetic form of blindness. One other type of therapeutic that may be, in the foreseeable future is again related to an easy delivery problem, which is disorders of the blood. So in this case, what you can do is basically take somebody's blood out and take the cells from the blood and actually edit them in a petri dish in a lab, and then change them the way you would like using CRISPR, and then put them back into the person. And so again, you're sort of circumventing that systemic drug delivery problem. And although that might seem sort of far-fetched or like science fiction, it's actually not the case. There are actually therapeutics that people are thinking about for HIV/AIDS. There are people that are thinking about for what's called immuno-oncology, and other types of immunological diseases that are really perhaps changeable when you try to target cells in the blood. So the eye and the blood, I think, sort of maybe in the foreseeable future. But just to throw a bit of optimism in there, as a scientist, in a long time, the pipe dream, and again, this is still a pipe dream, but the pipe dream is to have a one-shot injection that's curative for, let's say, hemophilia, or for some other genetic disease that we understand, like Huntington's or cystic fibrosis. Now, there are going to be a ton of hurdles that need to be overcome, and it might not ever happen. However, that's the pipe dream and the potential of this technology if we use it and develop it the right way.
I can also imagine that the challenge would be, how do you get a high enough dose for it to really work systematically? How do you harness the immune system to work with it? I guess these are all the kinds of problems you're working on in cell lines at this point, and modeling, is that right? And then we'll go to another question after that, James, but I just wanted to ask that question. Yeah,
that's a great follow-up, and yes, that is in fact correct. And in addition to that, there are other hurdles, like, again, not to harp on delivery too much, but it's pretty, I guess, easy to understand and agreeable. We can all agree on this, that if we're going in and delivering a drug that's going to actually edit the genome, and we want to do that in the heart for some heart disease, we don't want that going everywhere. We want to try to limit that and deliver it to the heart more so than the kidney or spleen or liver or wherever else we don't want it going. And so there are all of these hurdles that need to be overcome, including the ones you just mentioned, which are also good.
So how to make it selective. So of course, when people think of CRISPR, one of the things they think of right away is human embryos, right? How could it be used to go in and snip out or change expression of genes in development? And we know that there have been two groups that have worked with human embryo, well, cell lines at least, and then they were shut down. Melissa, do you have any thoughts about where that work is going, where the regulation is about what should be happening? How dangerous is that? It'd be good if you could sort of comment on where the community is with working with that, with human embryos and CRISPR.
I mean, I think, as we've said, that the CRISPR technology has changed how easy it is to do this, but it hasn't fundamentally changed what's going on. So I think there's been a discussion about this from the time that zinc fingers were first shown to be able to change genomes. And I think that we're still a really long way from it. I think it's always important to have the discussion started as soon as the possibility arises about what that is. But I think that honestly, there's still a lot we don't know about what specific changes to the DNA and how they're going to affect a developing organism, let alone a human, right? That there's interactions with environment and other things, and that there's a large number of very well-publicized disease-causing genes that might potentially be well-known and be causative, but I would say for the most part we don't have a handle well enough to make a designer baby at this point. So I think that that's not really on the forefront, but people are certainly talking about it, and I think that's really the most important thing, is to keep the discussion and to involve the public. So.
Do you think there should be some regulation up front before the science proceeds, or do you think it's too soon for that?
Yes, and I correct me if I'm wrong, others, but I believe that there are regulations, and so the work that has been done in human embryos has been on non-fertile embryos, and that's not been in the United States. So, you know, in the United States, there are very strict policies, is my understanding. As I said, I work on flies, but my understanding is there are very strict policies in place in the United States, and that these experiments that are making news are occurring abroad, where I don't think, you know, I don't know about international regulations on these sorts of procedures. So,
Keith, do you want to weigh in on this question? Do you have any thoughts about it as well?
Yeah, I think the only thing I would add is just, I mean, in the broader arc of the conversation, understanding the difference between the germline cells and non-germline cells, and so editing the DNA in my hand after I'm an adult isn't passed on to my progeny, where editing germline DNA not only affects me, but affects my progeny. So there are implications there that, as Melissa mentioned, they're not new implications, but I think they're more imminent because of the ability of CRISPR. And then the second thing that's people have been trying to grasp with regard to CRISPR is, so James talked about the drug delivery aspect of getting the CRISPR complex into the right cell, but even once it's in the right cell, while it's a specific system in terms of where it targets in the genome, it's not perfectly specific. And so we're still not fully appreciating how off-target editing might be happening in these organisms. I know there's a lot of effort in that area, because that, as we start thinking about therapeutics, understanding what the new side effects are, and these are now genomic inheritable side effects, is significant. So,
what do you mean by off-target editing?
So, so in Melissa's example, there was a particular gene that she wanted to target with GFP. However, the guide RNA sequence, which is what targets the CRISPR complex to where she wanted to put the GFP, it may target it to other parts of the genome that have similar sequences but not exactly the same sequence. And those events happen at a lower frequency than the target editing of it, but those off-target editing then could cause mutations that were not anticipated.
Thank you for that definition there. I appreciate it. That's obviously a big concern, that you be able to target very precisely with your systems. So now we have a question that has come in from Anne in New York about funding, and she's asking, can you talk about how funding affects your work? You're all relatively young, early-ish career investigators. Obviously, I'm going to ask you, do you see this as a hot area? Do you think the funding will increase? And how is funding affecting the direction you're going in with your research using CRISPR? And maybe which one of you, I didn't really target this. Melissa, perhaps, or any one of you have a strong feeling about this?
I mean, I'm welcome to talk about it, but my research is mostly funded in terms of its angle, in terms of asking questions about development. And for us, as I said, CRISPR is very much a tool that enables us to ask much more precise questions. And so I would say that from all that I've heard from colleagues and friends, that, you know, using CRISPR as a tool is becoming sort of not expected, but is very, very commonplace now in grant applications and things like that. I think, you know, there certainly is. It's almost moved so quickly from the level of super hot as a tool to expected as a tool that I think there's going to be a little bit of a correction, I think, back in terms of people recognizing that, you know, it's not as straightforward as, you know, that there are, when you're working in an organism or in a cell line or in a bacteria, that there are complications of working in a biological system that are going to make it not as trivial as it sometimes sounds on paper. But I think in general that there's going to be a strong push to use this technology because of its power.
It does sound like it's very much a tool. You're not getting funding for it as a tool. You're getting funding for interesting questions that you're addressing using this tool at this point, which is very interesting. We do have a question from Ewan at Stanford, Connecticut. It sounds to me like we're talking mostly about these advancements addressing diseases that will impact small numbers of people. Where does this research scale in humans without Congress slowing it down for years? Now, I did not hear that. I hear that it's still at the very basic research stages for disease. But let's have James or Keith address that, whichever one of you feels that you can address how this is impacting using CRISPR to address therapeutics for diseases.
Sure. I'll be happy to jump in here. So I think it's a little early to predict which diseases this is going to be useful for. People have made arguments that this is going to be useful for cancer. I can make strong arguments for CRISPR-based cancer therapeutics. I can make strong arguments against CRISPR-based cancer therapeutics. The same thing goes for heart disease and so on. So when you look at the diseases that affect a large number of Americans and a large number of people around the world, right now the science is still too early, in my opinion, to really predict, all right, we're going to be able to do X for this disease, which is a rare disease, and we're not going to be able to do X for this other disease, which is a more common disease. There are also more practical variables that influence this. So as an example, if you have a gene editing drug that could have off-target effects that we can't predict, and you have a rare disease where there's no alternative, there's no cure, there's no nothing, people may be more willing to try it out. But if you have CRISPR for headaches and you can just take an aspirin, people may be less willing to try it out. And so there are practical things, and that's an oversimplification, obviously, but there are practical things like what do we have available on the market now that are going to impact these things in ways that we don't really understand. And again, we can't predict, because in 15 years, the drugs that are available that are not gene editing drugs, just regular old drugs, are going to be different than the drugs we have now. So it's difficult to sort of predict the future like that at this point, in my opinion.
So I think Keith would like to add in too. Why don't you address this question and tell us about the value of studying this for, you know, therapeutics in general, or even just for basic research?
Yeah, James, and I felt like how you answered the end there, that the drugs we'll have in 10 years, which may not be CRISPR, but are going to be different than what we have today. I think we can't overemphasize how CRISPR is going to enable us to answer the fundamental questions that will help us determine what those drugs are in 10 years that will be, you know, even if the drug isn't a CRISPR-based drug, if we got to that solution was based on knowledge we discovered by CRISPR, is really nice. So I think that's great. The second thing I wanted to touch on is I think we are kind of thinking in the terms of, I think, like cancer and non-infectious disease and the type of people that might affect. But there is a fantastic work going on at the Gates Foundation, or funded by the Gates Foundation, I should say, trying to understand how to use CRISPR to target different gut microbes that cause diseases in the low-income countries, where things like cholera and diarrhea even can be deadly in children. And so those are some examples of things where antibiotics are just not a great tool to use, and we're learning that through antibiotic resistance. And so this gives us a new hook towards some of those type of infectious diseases that I think are very, very exciting.
Very exciting. It sounds like we're just beginning to get a sense of the sort of brave new world that CRISPR is opening up, both as a research tool, as Melissa is showing us, to help her tag proteins and to understand and trace them on their journey through development, so illuminating basic biological processes to actual drug delivery systems and all sorts of ways of maybe thinking about disarming microorganisms around us. Very cool. So I'm going to close on behalf of the Alumni Association. I want to thank you for tuning into this faculty forum online, Alumni Edition. A special thanks to our MIT alumni panelists from Northwestern, Wisconsin, and Georgia Tech. We didn't get to all the questions submitted, but our panelists will soon see them all. You can tweet about today's chat using the hashtag MITBetterWorld, or send any follow-up questions or feedback to alumnilearn@mit.edu. Thank you to each of the panelists, and thank you to you out there watching. Thanks so much.
Thanks again for joining us. For more information on future MIT Alumni Association productions, please visit our website.